Welcome back to Acing AP Biology. In this lesson, we will be going over how DNA is packaged into chromosomes. Let's first look over the differences between prokaryotic and eukaryotic chromosomes. Prokaryotic chromosomes are circular and are much smaller than eukaryotic chromosomes. They are held in the nucleoid region. Not many proteins are associated with it. It is anchored to the plasma membrane. Eukaryotic chromosomes are linear. During interphase, they are tangled and uncondensed in the form of chromatin. Therefore, there is an intricate DNA packing system condensing chromatin into chromosomes. Genetic material is in the form of chromosomes for mitosis. There are two forms of interphase chromatin, heterochromatin and Eurochromatin. Heterochromatin is visible under the light microscope as it is very dense, so no DNA transcription can take place. This allows for the regulation of gene expression. An example of heterochromatin are Barr bodies, which result from X chromosome inactivation in female mammals, as we talked about in Unit 10. The formation of Barr bodies involves the methylation of DNA. Euchromatin is the less condensed form of interphase chromatin, allowing for transcription to take place. The addition of acetyl groups to DNA through acetylation promotes gene expression. Now let's look over the stages of DNA packing. The first stage of DNA packing involves nucleosomes. Nucleosomes are often called beads on a string and are a 10 nanometer fiber. Histones are positively charged proteins that are bound strongly to DNA, which is negatively charged, and aid in packing. A nucleosome is composed of a protein core made up of 4 pairs of different histone proteins wrapped twice by DNA. Therefore, the nucleosomes act as the beads, and the strings are the linker DNA that connect nucleosomes with each other. The next stage of packing begins with the attachment of an H1 histone protein near the nucleosome. The next level of DNA packing is called the 30 nanometer chromatin fiber. This process is conducted by H1 protein, histone tails, and linker DNA between nucleosomes. Afterwards, looped domains begin to form. A loop of the 30 nanometer fiber binds itself to a scaffold protein, and the looped domain structure forms. Next, chromatin condenses even further, forming a 300 nanometer fiber. Eventually, it condenses far enough to form chromosomes, and now is ready for mitosis. Here is a diagram displaying the stages of DNA packing. First, positively charged histones bind to the negatively charged DNA, and nucleosomes form, with 4 pairs of histone proteins surrounded twice by DNA. Linker DNA holds consecutive nucleosomes together. Then, with the help of the H1 protein, the 30 nanometer fiber forms, which is the next stage of DNA packing. Then, the 30 nanometer fibers loop around a scaffold protein, forming the loop domain. Loop domains condense even further to form the 300 nanometer fiber, which eventually gives rise to a chromosome. That's it for this lesson. In the next video, we will introduce the topic of DNA transcription. See you then. For more bio content, hit subscribe.